Cytoplasmic dynein exerts a pulling force on the centrosome through its -N directed movement, which enables centrosome separation during mitosis. Dynein present at the kinetochores regulates microtubule kinetochore attachment, meiotic chromosome motion, and spindle assembly checkpoint signaling. Several protein complexes help in the recruitment of the dynein-dynactin complex at the cortex. Cortical dynein pulls the meiotic spindle apparatus to position it at the center of the cells. Cytoplasmic dynein is also found to localize at the meiotic spindle poles and helps in the transport of paracentrin, gamma-tubulin, and nuclear meiotic apparatus protein (NUMA) to the centrosome that controls centrosome maturation and spindle assembly. Axonal and cytoplasmic dynein 2 is present in cilia and flagella, where it functions in retrograde transport to ensure axoneme structure maintenance. Experimental evidence showing the role of -N directed motor proteins during meiotic spindle assembly. RNAi-mediated gene knockdown of the dynein heavy chain DHC64C NCD, or double RNAi of both motors was conducted in Drosophila S2 cells. Immunofluorescence studies were done on these cells to examine pole focusing of metaphase spindles. The lateral spread of the kinetochore -Ns termed K-fiber distance, and the distance between the centrosome and the base of the K-fibers centrosome to K-fiber distance were measured. Chromosome detachment, as represented by an increase in centrosome to K-fiber distance, was observed upon dynein heavy chain knockdown. Interestingly, NCD depletion by RNAi did not alter the centrosome to K-fiber distance. In contrast, RNAi of NCD showed a strong phenotype on K-fiber focusing. The effect was more pronounced upon double knockdown of DHC64C and NCD than each single RNAi. These results suggest that NCD has a major role in K-fiber focusing, while dynein primarily facilitates chromosome attachment in S2 cells. Cilia and flagella, structure and movement. The evolutionarily conserved cellular appendages, cilia plural of cilium and flagella plural of flagellum, are motility and sensory organelles made up of microtubule MT bundles. The flagellum derives its name from the Latin word meaning whip, while the cilium is Latin for eyelash. They vary primarily in terms of their number and position on a cell. The cilia are shorter and seen at higher numbers, usually distributed across the cell membrane. The flagella, on the other hand, are much longer and fewer in number and often seen at restricted sites of the cell. Based on their function, cilia are of two types. The motile cilia, as the name suggests, aid in movement and is found on cells like the sperm or on multiciliate epithelium. The non-motile variant of the cilia is the primary cilia, also known as the sensory cilia. The primary cilia, unlike their counterpart, are much shorter and are involved in other functions like sensing extracellular cues. A classic example is the presence of primary cilium along the nasal epithelium, aiding in sensing changes in the extracellular cues. Type example: cilia motile lining of trachea, intestine, fallopian tubules, non-motile sensory organs. Nose flagella bacterial E. coli archaeobacteria eukaryotic sperm cells table 4.4 types of cilia and flagella. Structure. The flagellum of a prokaryotic cell is made up of three parts: a basal body, a hook region, and a filament structure. The basal body is the region that anchors the flagellum to the cell membrane. The hook region holds the structure in place by penetrating the cell wall and connecting the basal body to the filament. The filament structure is entirely made up of a protein called flagellin, primarily made of the amino acids proline, histidine, cysteine, and tryptophan. The eukaryotic flagellum, unlike its prokaryotic counterpart, is a complex arrangement of microtubules made up of two regions, the basal body and the axoneme. Figure 4.50 The structure of a prokaryotic flagellum. The schematic representation of a prokaryotic flagellum. The basal body connects to the cell and plasma membrane. The hook extends from the membrane and holds the structure in place while the filament structure is made up of a protein called flagellin. Axoneme. The axoneme is the region that extends into the exterior environment. Irene Manton in plants, Don Fawcett and Keith Porter in animals were the first to describe the structure of the axoneme. Basically, ciliar flagella are columns of microtubules covered in a membrane. Each microtubule is, in turn, made up of tubulin heterodimers that polymerize into protofilament strands. The axoneme, which is the core structure of cilia and flagella, is made up of two central microtubules enclosed in a central sheath. These are cylindrically surrounded by 9 doublets of microtubules. Hence, referred to as the 9+2 arrangement of microtubules. The central pair of microtubules, also called the central pair apparatus (CP), is made of two singular and complete rings of 13 microtubule protofilaments. Each outer doublet is formed of one complete ring, called a tubule, and the other doublet of 10 protofilaments extend from the A ring toward the center, while necks and bridges connect the doublets together. An outer and an inner dynein arm extend from the A ring. Basal body. The centriole, when supporting a cilium, is called the basal body. Similar to the centriole, the basal body is the region from which the axonal microtubules originate. The basal region, unlike the axonal MTs, has a 9+0 arrangement, with 9 triplets of microtubules at the periphery and no central microtubules. The structure of the primary cilia is slightly different from that of the motile cilia in that they do not have the central microtubules and the dynein arms. Eukaryotic cilia and flagella. Most eukaryotes do not have a true flagellum, with the exception of the sperm. The sperm requires a flagellum to swim towards an egg. Eukaryotic cilia and flagella have the same structural features but are not similar to their bacterial counterparts. The bacterial flagella are very different from their eukaryotic counterpart. They lack microtubules or dynein, and are merely repeated units of long, rigid filaments composed of a protein called flagellin. A rotary motor present in the bacterial cell wall helps rotate the flagella to mediate movement without any ATP utilization. Another major difference is the way the flagellum moves. The prokaryotic flagellum moves in a rotatory fashion using a rotatory motor, while the eukaryotic flagellum moves in an undulating fashion using the sliding filament principle of microtubules. Called the B-tubule. The 9+2 arrangement of the axoneme is a highly conserved one ranging from protozoans to humans. The doublets are connected by inelastic proteins called nexin that form interdoublet bridges. Two sets of axonal dyneins bind to the A-tubule, the outer and the inner dynein arm. A set of radial spokes aids in connecting the A-tubule of the outer doublet microtubules to the central microtubule and thus aids in spacing the outer tubules to the central ones. Figure 4.51 The structure of ciliary and flagellar axoneme. The schematic representation of the axoneme of a protist. The central sheath enclosing two central singlet microtubules, surrounded by 9 outer doublets of microtubules, enclosed within the outer plasma membrane. The doublet microtubule is comprised of an outer complete ring and one incomplete ring B radial spokes. Figure 4.52 The prokaryotic and eukaryotic flagellum. The prokaryotic flagellum moves in a rotatory fashion while the eukaryotic flagellum move in a wave-like pattern. The structure of the axoneme is crucial in mediating the ciliary and flagellar motion. The delicate interplay of dyneins walking along with the microtubule results in a highly coordinated movement of the ciliar flagellum. We will now look at each part in detail. Axonal microtubules. The axonal MTs share a lot in common with the cytoplasmic MTs discussed earlier. However, the axonal MT is a highly stable structure and has been shown to be resistant to no-cortisol and cold, agents that mediate complete disassembly of cytoplasmic MTs. The axoneme comprises the core of the cilia and flagella. The axoneme is made up of microtubules arranged in a 9+2 pattern, an outer ring of 9 doublets, and a centrally arranged pair of single complete microtubules. Each microtubule is made up of a repeating array of alpha and beta tubulins arranged in turn into protofilaments. The doublets are formed of one complete 13 and one partial microtubule 10. The microtubules are oriented with one end closer to the cell, called the minus end, while the opposite end is called the plus end. The microtubules extend continuously along the length of the axoneme, supported by accessory proteins that link the microtubules together. A few exceptions to this include the eel sperm, diatom gametes, and the nodal cilia in mouse embryos, where the flagella lack the central pair of microtubules. A, B. Figure 4.53 The microtubule arrangement in the flagellum. A. The ultrastructure of the MTs vary across the flagellum, with a 9+2 double and 2 singlets arrangement in the axoneme, a 9+0 arrangement, 9 outer triplets in the transition fiber, and a 9+0, 9 outer triplets arrangement in the basal body. B. Microtubules can be arranged as singlets of one complete ring with 13 protofilaments, as doublets, with one complete ring of 13 filaments and one incomplete ring with 10 protofilaments, and as triplets in basal body with one complete and two incomplete rings. Motor proteins in the axonal MTs, like their cytoplasmic counterpart, the axonal MTs also bind motor proteins which help mediate specialized transport of cargo called the intraflagellar transport system. The axonal MTs form tracks on which kinesis 2, the plus end directed, and dynein, the minus end directed motor protein, carry cargo along the MTs. Since translation does not occur in the cilium, the protein composition of proteins like tubulin is determined largely by the motor protein-driven transport of cargo between the cytoplasm and the cilium. Experimental evidence. Structure of axoneme microtubules. The primary cilia are slightly different from the modal cilia, in that their axoneme do not have the central pair of MTs 9 plus O. The eukaryotic flagella thus exists as either a 9 plus 2 axoneme MT for motility or a 9 plus 0 axoneme MT mediating sensation and signaling. An interesting question would be to understand if and how these two kinds of axoneme MTs could interchange post-assembly. Researchers from the University of Oxford set out to explore this phenomenon in the unicellular eukaryotic parasite Leishmania mexicana. The L. mexicana parasite forms a 9 plus 2 modal cilium in the promastigote life stage while it adopts a 9 plus 0 modal cilium format in the amastigote stage. The 9 plus 2 structure of the axoneme highly resembles the human modal cilia while the 9 plus 0 format structurally mirrors the mammalian primary cilium. In vitro models of L. mexicana were adopted to analyze the length, structure, and biochemical changes. Transgenic Leishmania cell lines expressing fusion proteins of enhanced green fluorescent protein EGFP and axonal proteins were used to study the transition between the promastigote to amastigote stages. Figure 4.54 The differential arrangement of MTs in the axoneme. The ultrastructure of MTs in the axoneme of a modal and non-modal cilium varies. MTs are arranged in a 9 plus 2 format in the modal cilium whereas the non-modal cilium has a 9 plus 0 arrangement. From the results, we can clearly see that the pre-existing modal cilium exhibits a great level of plasticity allowing it to remodel its structure to suit a lifestyle as a pathogen. Cells in the transition of promastigote and amastigote stage can easily transit from a 9 plus 2 to a 9 plus 0 cilium by degradation of the central microtubules. The 9 plus 0 structure can even form by de novo growth. This is also evidence of the fact that the basal body is not committed toward any one axoneme transformation. Post-translational modifications in the tubulin. The long-lived tubulins, which are the basic subunits of the microtubules, undergo several post-translational modifications (PTM). Among the tubulins, the alpha tubulin is usually unmodified, but the beta tubulin is seen to be modified in a subset of microtubules. The beta tubulins are detyrosinated, polyglycolated, and polyglutamylated. Glutamylation is central to key ciliary functions like assembly, motility, and stability. Loss in the enzyme tubulin tyrosine ligase, like TTLL, that mediates glutamylation results in motility defects in sperms. On the contrary, excessive glutamylation can result in destabilizing and shortening of the cilia. An important PTM of the alpha tubulin is the acetylation of K40. Acetyl K40, a marker for long-lived microtubules, marks both the central and outer microtubules, unlike most PTMs that are associated with the outer tubules. Axonal dyneins. The axonal dynein, ciliary dyneins, which is the second branch of the dyneins, is composed of monomer, heterodimer, or heterotrimer motor containing heavy chains. The dyneins are arranged at regular intervals along the length of the microtubule doublets. Dyneins help to form bridges between two neighboring doublets in the axoneme, efficiently mediating the sliding movement of microtubules by generating the force required. These molecular motors are very fast and can move microtubules at a rate of 14 micrometers per second on glass slides. ATP hydrolysis triggers the dynein arms to walk over the neighboring axoneme. Figure 4.55 Dynein activity. The regulation of dyneins are regulated both along the length and also across the circumference of the axoneme. The active dyneins are marked in red and inactive ones are marked in yellow. All the dyneins do not get activated at the same time. In order to generate a bend, the movement of the dynein must be regulated not only along the length of the tubule but also across the circumference of the axoneme. When one side is active, the other side remains inactive. There is also a dynein regulatory complex (DRC) that seems to be a key regulator of dynein activity. However, a detailed structure of DRC was not available. Researchers from the Brandeis University have been able to shed some light on the structure of the DRC through cryo-electron tomography of axonemes from Chlamydomonas reinhardti. The DRC has a total length of approximately 50 nanometers with two domains, a base plate BP and a linker domain. The BP is attached to the lower side of the A ring. The linker domain extends from the A ring and connects to the B ring of the neighboring doublet. The linker domain has at least three protrusions, the L1, L2, and the outer dynein interdynein OID linker. Structurally, since the DRC is the only connecting link between neighboring doublets, it is suggested that perhaps DRC is the nexon linker itself and is, therefore, often referred to as the nexon dynein regulatory complex (NDRC). Beating of cilia and flagella. Signal to mediate motility pass from the central MT pair complex through the radial spokes, the inner dynein arm and the dynein regulatory complex. The directionality of the dynein movement is maintained simply by the fact that dyneins are -N director motor proteins. The binding of dynein to the A tubule is not ATP dependent. However, the walking of the dynein along the beta tubule is ATP dependent. The axonal movement is mediated by the controlled sliding of the outer microtubule doublets. Several models for understanding the mechanism behind the dynein-dependent sliding of the outer and inner microtubules have been made. The primary mechanism revolves around the regulated manner in which the dynein motor proteins are activated to walk along the MT of the neighboring outer doublet. To understand this, we need to have a better understanding of the dynein walk along the MT. Dynein walk. Dynein is able to walk along a microtubule track in a processive manner, taking a step each on its head domain. A detailed description of the structure of dynein is given earlier. The coordinated fashion in which the dynein walk has been termed the gating mechanism. A very similar mechanism is followed by the kinesin motor protein, also described in. Just like the kinesin protein, the dynein too requires head-head communication and also has an intermediate state where one head domain is tethered while the other head domain is detached. The gating mechanism describes how each head domain waits till its partner head domain opens a gate, allowing the previous head domain to proceed to the next step. This can be described either as a polymer gate, defining the rate at which the motor head binds or releases from an MT, or as a nucleotide gate, which defines the rate of the ATPase cycle that activates the head domain. Together they regulate the rate of the ciliary or flagellar beat. Figure 4.56 Schematic representation of the dynein structure. The dynein motor protein is composed of two main heavy chains and several associated chains. The coiled coil stock domain has a microtubule binding domain MTBD. The catalytic motor domain contains several AAA ATP binding sites, 1 to 4, but the 5, 6 and the C-terminus C do not have the same features. A linker domain connects the motor and dimerization domains. The dynein light chain is bound by several associated chains, LIC, light intermediate chain, IC, intermediate chain, roadblock, TC-TEX1 and LC8, light chains. A model by Alonso et al. Tries to explain how this movement proceeds. The model describes a state where one head domain is bound to the MT while the other detached head waits near the bound head towards the plus end. This creates a polymer gate, which is opened when an ATP binds to the bound head, causing a conformational change resulting in the detached head rebinding onto the MT, causing the dynein to step forward. When a signal for movement is generated, it activates the motor head domain of dynein. As a result, the dynein initiates to walk along the neighboring microtubule, forcing the microtubule to slide across the microtubule to which the dynein head is attached. However, what should have been a sliding action results in a bending motion as the microtubules are not free but attached to each other by separate linking proteins called the nexon. Repeated bends can result in a beating movement. The dynein-driven movement of both the cilia and flagella can either result in the movement of the cell through a fluid or, when present on cells arranged in a group, can result in the movement of fluid over the cells. Regulation of sliding motion has been shown to be mediated by phosphorylation of the intermediate chain IC138 and can also respond to changes in calcium concentration. Specifically, the centrin in the DRC and the calmodulin in the radial spokes function as calcium-binding proteins that can sense free calcium and respond accordingly. Properly functioning cilia and flagella are essential for the normal functioning of the body. Situs inversus is a condition in which a person's internal organs are reversed. Mutations in genes encoding the dynein chains can result in abnormal axoneme characterized by dysfunctional outer or inner dynein arms, radial spoke or microtubules. It is characterized by respiratory infections and male infertility. Experimental evidence. Ciliary flagellar synchronization. An individual cilium or flagellum can beat according to its own rhythm. However, correct ciliary orientation and coordination are crucial when several cilia are present together on the surface of cells. A typical example is the movement of an egg cell along the fallopian tube. The process requires the cilia lining the membrane to beat in a coordinated fashion. Although most ciliopathy are associated with the structure of the cilia, a few variants like the primary ciliary dyskinesia (PCD) are associated with the lack of oriented cilia. The earlier belief was that a central pacemaker within the cell facilitated the synchronous beating of cilia in the same cell. However, evidence of synchronous beating of the flagella of sperms that swam together indicated that there could be other means of regulating the rhythm. Studies from the University of Cambridge have demonstrated how hydrodynamic interactions play a key role to mediate flagellar synchronization. Somatic cells of the green alga, Volvox carteri, with different beating frequencies were studied by high-speed imaging. For the experiment, individual biflagellate cells were isolated from a ball box colony and inserted into glass chambers containing a standard ball box medium. Micropipettes were used to capture and orient these cells such that their flagellar beating planes coincided with the focal plane of an inverted microscope. High-speed video cameras were used to capture the synchronization pattern. Figure 4.57 Synchronous beating of the cilium flagellum. A. The image depicts several green alga ball box colonies. Each cell in a colony has an individual flagellum. B. The experimental setup of setting two such isolated flagellum at a given distance L2. Understand how hydrodynamic forces can influence the rhythm of beating between the flagellum. C. A possible contributor to synchrony could be the ability of the motor dynein to sense the external cues. Synchronous beating of cilia is suggested to be dependent on the distance. A distance of more than 10 micrometers between the flagellum cilium could easily disrupt the synchrony. A spatial dependence for synchrony was deciphered, where lesser spatial separations between the flagella resulted in increased synchrony. When close enough, the flagella was seen to beat at the same rhythm for thousands of beats. They reported that the synchrony is perturbed beyond 10 micrometers of distance. Interestingly, fluid flow produced by a beating flagellum is sufficient to mediate the synchronous movement of a nearby flagellum. The ability of the motor proteins to sense external cues is critical in maintaining the beat pattern. Microtubule sliding. As mentioned earlier, in the absence of the linker protein nexin, the movement of dynein on the MT would result in the sliding of MT over each other instead of a bend. As the dynein walked towards the minus end, it would slide the adjacent MT towards the plus direction. Bend. In normal flagellum, dynein causes microtubule bending. In isolated doublet microtubules, dynein produces microtubule sliding. AB plus ATP. A. As the dynein walk across the neighboring microtubule MT towards the minus end, the adjacent MT is pushed towards the plus end mediating an MT sliding action. B. In the flagella, the MTs are held in position by linking proteins and so when the dynein walks along the MT, the stress generated results in a bend as the MTs are bound. Unlike the structure in the cilia and flagella, the MT tracks are not always stationary, implicating that the MT sliding movement is also an essential feature of the MT. A classic example is the formation of cellular processes where MT bundles slide along each other. However, studies have proven that, unlike what was earlier believed, kinesis 1 is attributed to mount sliding rather than dyneins. Intermediate filaments. Early understanding of the intermediate filaments IFS came from the work of Howard Holzer and his group when they detected smooth and uniform filaments along with the actin and myosin filaments in muscles with a size that was intermediate to both actin and myosin at 10 nanometers. Further, with the discovery of the 10 nanometers filaments, even in the non-muscle cells, these filaments came to be called the intermediate filaments, also being intermediate in size compared to the other cytoskeletal proteins, the microtubules and microfilaments. If are a set of key intracellular proteins that are responsible for providing the structural framework and integrity to the cell and tissue. These highly diverse form of the cytoskeleton is encoded by around 70 different genes in the human genome, expressed in a very cell and tissue specific manner. Intermediate filaments, structure and function. The basic structure of an IF subunit monomer consists of a conserved central alpha helical rod and a non-helical domain at either end. The helical domain contains haptid repeats tilde operator 352 residues with subdomains 1A, 1B, 2A, and 2B. The helical rod domain is flanked on either end by a non-helical, conserved amino head terminal or a carboxy tail terminal. The head and tail domains can vary considerably in their structure and length, contributing to the wide heterogeneity seen in the family. The helical rod domain is responsible for the self-assembly of the filaments forming a coil-coil dimer, while the end terminals mediate the interaction with other filaments. A. B. Figure 4.59 Structure of an intermediate filament. The schematic is a representation of the secondary structure of an IF, with a central alpha helical rod and subdomains 1A, 1B, 2A and 2B amino and carboxy terminals flanked by the head and tail domains. B. Different monomers of the proteins and their respective structural details. The alpha helical repeats can form a coil-coil structure with another monomer forming a parallel dimer, where the N and C terminals of the dimer are aligned. The dimers formed could either be homodimers or heterodimers. These dimers further form a staggered tetramer by binding in an anti-parallel fashion resulting in a nonpolar structure. These tetramer structures are considered the basic soluble subunits of the filaments. 8 tetramers were further twisted into a unit-length filament. Each filament is thus an association of 32 alpha helical coils and ranges around 60 nanometers. The unit-length filament can further aggregate in an end-to-end manner to form long rope-like filaments of over 240 nanometers. However, post-aggregation, the filament undergoes compaction, where the filament is shrunk to a final size of around 10 nanometers. Unlike the microtubules and microfilaments, the IFs assemble in a nucleotide-independent self-assembly manner. Figure 4.60 Intermediate filament polymerization. Type 4. Type 4 is composed mainly of neurofilaments NF and alpha internexin proteins which are expressed in neurons. The NFs consist of NF light, NF medium and HF heavy, which are aligned as strict heteropolymers at a molar ratio of 5, 3, 1. NFs are present specifically along the axons of neurons. Since the NFs are associated with the axons and require dynamic changes corresponding to the growth of the axon, there is a direct relation between the diameter of the axon and the expression of the NF gene. They are responsible for providing sufficient strength and stability to the neurons. Type 5. The inner nuclear membrane is associated with a meshwork of IFs called nuclear lamins coded for by the type versus IF gene. The nuclear lamins are composed of A isoforms include lamin A and lamin C and B type lamins. The lamins are attached via lamin binding proteins to the inner nuclear membrane. The lamins have a regular tripartite structure with an alpha helical central domain and amino and carboxy terminal globular domains. Additionally, the lamins have a nuclear localization signal sequence between the carboxy end and the central rod domain and at the carboxyl terminal tail domain, a binding site for chromatin and a CAX isoprenylation signal sequence. Nuclear lamins are the key proteins that confer mechanical stability to the nucleus. Type 6. A smaller group of IFs, composed of faconin, CP49 and phylincin, are present in the I-lens. These cytoplasmic, beaded filaments do not follow the classification criteria and are thus put as a separate group. Some references classify them as the non-standard type 4 IF proteins. The helical rod regions of the IF monomers coil around other IFs forming dimers. The dimers align anti-parallelly to form staggered tetramers. Repeats of tetramers form a unit-length filament, approximately 60 nanometers, which further aggregate in an end-to-end manner to form the larger filaments, and GT, 240 nanometers. Intermediate filaments proteins. Based on their structure and nucleotide sequence homology, IFs can be classified into 6 types. Type 1 and 2. The type 1 and 2 genes code for 2 types of keratins, acidic and basic keratins, respectively. Keratins are considered the most diverse IF protein, with around 54 distinct types of keratins, specifically 28 type 1 and 26 type 2 keratins. Heterodimers of keratin are formed by copolymerization of an exclusive and equal mix of acidic and basic keratin proteins, such that a dimer is composed of an acidic and a basic monomer each. Cytoplasmic keratin fibers are present in all epithelial cells. Keratins are either hard, like those in the hair and nails, or they can be soft keratins, also called the cytokeratins, present in the epithelial cells of the skin. Keratins anchor at desmosome or hemidesmosome sites to contribute to the mechanical stability of the cell. Detection of the type of keratin has been extrapolated in cancer studies to understand the origin of these epithelial cells. Type 3. Among the type 3 IFs are vimentin, desmin, glial fibrillary acidic protein, GFAP, parafferrin, etc. Vimentin fibers can homopolymerize into 10 nanometers thick filaments and are seen mainly in mesenchymal cells. Desmins are found around the Z disc of skeletal, cardiac, and smooth muscle cells, stabilizing the muscle fibers. The GFAP proteins are found in the glial cells and the parafferins in the peripheral neurons. Localization type name size. KD. Assembly. Group cell tissue distribution. Cytoplasmic acidic keratin K9K20. 40 to 64A epithelial cell, hair. 2 basic keratin K1K8. 52 to 68A epithelial cell, hair. 3 vimentin 55B mesenchymal cells, fibroblasts, endothelial cells, astrocytes. Desmin 53B muscle. GFAP 52B astrocytes. Parafferrin 54B peripheral neurons, IBNFH 110B CNS neurons, NFM90B CNS neurons. NFL 61B CNS neurons. Alpha internexin 61B CNS neurons. Cinnamon CNS neurons. Nestin CNS neurons. Nuclear B. IFs are highly stable as the major roles of IFs do not require them to be dynamic. However, other roles of IFs, like those involving momentum, during MET transitions and nuclear lamins during mitosis, require them to undergo phases of polymerization and depolymerization. Compared to the microfilaments and the microtubules, very little is known about the assembly and disassembly of IFs. The phosphorylation of proteins is suggested to be the mechanism behind the regulation of disassembly. For example, during mitosis, cyclin-dependent protein kinase CDK1 CDC2 kinase phosphorylates nuclear lamins resulting in the disassembly of the nuclear membrane. While dephosphorylation of lamins, post-prophase, enables the reassembly of the nuclear lamins. Several of the IFs, including vimentin, GFAP, etc., when exposed to phosphorylating agents like protein kinase C, PKC, or CAMP-dependent protein kinases, under in vitro conditions, would easily disassemble. The phosphorylation site is often present on the non-helical amino head domain, implicating a very site-specific phosphorylation of IFs. Figure 4.61 Organization of IF governed by phosphorylation. Phosphorylation by protein kinases at the amino terminal domain in a site-specific manner of IF induces disassembly, while a dephosphorylation by protein phosphatases results in assembly of the IF. The phosphorylated or dephosphorylated state determines the state of the IF subunits in soluble or insoluble filaments. Further studies on vimentin during mitosis revealed that the phosphorylation pattern is not only site-specific but is also regulated in a very spatiotemporal manner by cooperative phosphorylation of different kinases. Figure 4.62 Cooperative phosphorylation mediated by different kinase. Phosphorylation of S55, S82, S71, and S72 in a site-specific manner, mediated by the cooperative phosphorylation of specific kinases, namely the cyclin-dependent protein kinase CDK1, polo-like kinase 1, PLK1, Rho kinase, and the Aurora B kinase. Phosphorylation of the vimentin during the prometaphase is mediated by CDK1 at serine. 1. S55. The metaphase sees the polo-like kinase 1, PLK1 being activated by the phosphorylated S55, and in turn, the activated PLK1 phosphorylates the S82. While at the anaphase, S71 and S72 are specifically phosphorylated by a separate set of kinases, the Rho kinase and the Aurora B kinase, respectively. Intermediate filaments in cell junctions in a polarized epithelial cell. Cell junctions can be classified as occluding junctions, anchoring junctions, and communicating junctions. Among these, the IFs play a role in the anchoring junctions, mediating both the cell-cell and a cell matrix interaction. The desmosomes connect the IF between cells, while the hemidesmosomes connect the IFs to the extracellular matrix. Structurally, both the desmosome and the hemidesmosome basically consist of a transmembrane adhesion protein that mediates contact either with an extracellular ligand on another cell or the matrix, respectively, an intracellular anchor protein that connects the adhesion protein and the IF, and finally the IF. Anchoring junctions mechanically attach cells. Cell-cell junctions, adherens junctions, cadherin, E-cadherin, actin filaments, desmosomes, cadherin, desmocoline, desmocoline, intermediate filaments, cell matrix junctions. Focal adhesions, integrin, actin filaments, hemidesmosomes, integrin, alpha, beta 4, BP180, intermediate filaments, table 4.6 types of anchoring junctions. Figure 4.63 Cell junctions. There are several kinds of cell adhesion proteins. The four main cell adhesion junctions are tight junctions, adherens junctions, desmosomes, and gap junctions. The cell matrix interactions are mediated by hemidesmosomes and focal adhesion junctions. Desmosomes. Desmosomes serve as the anchoring sites for IFs. The desmosomes mediate the interaction of IFs from adjacent cells, and this communication can extend across several cells of a tissue. The IF attached to the desmosomes is usually keratins in epithelial cells and desmins in muscle cells. Desmosomes are made of transmembrane cadherins, anchoring proteins, and IFs. The transmembrane adhesion proteins like the desmocoline and desmocoline belonging to the cadherin family aid in holding the plasma membrane together. The cadherins, via their extracellular domain, bind to the cadherins of the adjacent cell in a Ca2+ dependent manner and to the cytoplasmic plaque via their cytoplasmic domain. The cytoplasmic plaque is a complex of intracellular anchoring proteins, namely the plakoglobin and desmoplakin. The plakoglobin is bound to the cadherins, further bound to the desmoplakin, and finally to the IF keratin. Figure 4.64 Structural components of desmosomes. Image 8 represents the electron micrograph of desmosomes between two epithelial cells in the intestine of a rat. B. Electron micrograph showing a detailed structure of a desmosome between two epithelial cells of a developing newt. C. A desmosome made of anchoring proteins forming a plaque on the cytoplasmic side. The plaques attach to the keratin bundles. Cadherins, the transmembrane proteins, attach to the plaques on the cytoplasmic side and to cadherins of the adjacent cell via their extracellular domain. D. The desmocoline and the desmocoline cadherin family bind to the plakoglobin gamma-catenin on the cytoplasmic side, which in turn binds to the desmoplakin that binds the keratin IF. Hemidesmosomes. Hemidesmosomes, also called the half desmosomes, are very similar to their desmosome counterparts. However, instead of cell-cell interaction, they mediate a cell matrix interaction. They connect the basal surface of the cell to the extracellular membrane of the basal lamina. Here the integrins play a role in regulating the adhesions. The extracellular domains of integrin bind to the laminin protein in the basal lamina, and the intracellular domain binds to an anchoring protein called the plectin. The plectin finally interacts with the IF keratin. The integrins are transmembrane heterodimer proteins that mediate both adhesion and signal transduction. A classic example is an alpha-6 beta-4 integrin, which is a laminin receptor. The cytoplasmic interaction of the integrin is mediated via the cytoplasmic domain of the beta-4, approximately 1,000 residues. The cytoskeletal linker, plectin, from the plakin protein family, is a direct connection between the integrin and the keratin filaments. Other associated proteins include BPAG1E, also known as BP230, BP180, known as collagen 17, and the tetraspanin CD151. Figure 4.65 Structural components of hemidesmosomes. A. The hemidesmosomes help attach the basal keratinocytes to the basement membrane. B. Schematic representation of the hemidesmosome complex. Adhesion begins with the binding of the extracellular domain of the integrin alpha-6 beta-4 to the laminins in the basement membrane. The beta-4 domain extends into the cytoplasm and interacts with plectin, which in turn is a link between the integrin and the keratin. C. Representation of the binding of plectin to the fibronectin type 3 FNII1 and FNII2 domain of beta-4. Microtubule intermediate. Filaments. Microfilament actin. Filaments. Size 25 nanometers, 10 nanometers, 8 nanometers structure, hollow cylinders, rope-like fibers, helical polymers. Monomers, tubulin type 1, type 5 actin. Energy GTP, nucleotide independent. ATP. Polarity, polarity, lax polarity, polarity. Motor protein kinesin and dynein myosin. Localization in the cells. Cytoplasm, cytoplasm and nucleus. Cytoplasm. Key functions: maintain cell shape, mediate motility. Mitosis spindle. Fibers, localization of organelles. Transport tracks for motor proteins. Maintain cell shape, anchorage sites for chromosomes, nuclear lamins, mechanical. Strength, form, thick appendicature, nail strength and shape to the cell cortex. Motility, lamellipodia, filopodia. Contraction, muscle stress fibers. Diseases, disorders, associated ectopic pregnancy due to non-functional cilia in the fallopian tube, male infertility, epidermolysis bullosa simplex. Lou Gehrig's disease, muscular dystrophy. Table 4.7 comparative note on cytoskeletal proteins. Cancer cell. The development and causes of cancer. Cancer is a complex genetic disorder caused due to multiple genetic alterations, which lead to uncontrolled cell division, leading to the establishment of pathological masses of cells called tumors. Hence, it is a disease of uncontrolled meiotic cell division. Cancers are capable of invading nearby tissues and also spreading to other parts of the body through the blood and lymph systems. It is not a single disease, but comprises a large group of diseases and can originate in almost any organ or tissue of the body. Cancer is one of the most prevalent diseases globally and is responsible for a great amount of morbidity and mortality among the human population. Surveys conducted by global agencies have shown that it is increasingly a global health issue. The WHO has projected an alarming The WHO has projected an alarming increase in the number of cancer incidences and cancer-related deaths per year by 2035. In view of the importance of this disease, the 4th of February has been marked as World Cancer Day, with the purpose of raising awareness of cancer, its prevention, detection, and treatment. The human race's struggle with cancer dates back several centuries. Since the time it has been acknowledged as an ailment of significance, several scientists have been incessantly working towards understanding the causal mechanisms and developing therapeutics in an attempt to remedy the disease. Human cancers include more than 200 different diseases, affecting various cell and tissue types. The meiotic cell division is a key process in the growth and development of an organism. During development, cells divide and differentiate into specific tissue types, which are further organized into organs, organ systems, and the whole organism. In normal conditions, the balance between cell birth and cell death is strictly regulated in response to growth signals, growth-inhibiting signals, and death signals. Certain adult tissues, such as intestinal epithelium, blood cells, skin cells, etc., retain the ability to divide as a continuous renewal of the tissues is required. However, in other cases, the cells in adult tissues do not normally proliferate except during healing processes. In certain abnormal situations, these cells may acquire the ability to divide uncontrollably by overriding the pre-existing regulations and form masses of cells. The term neoplasm is used to define such a persistently growing mass of abnormal cells, and the word tumor is more commonly used as an alternative to neoplasm. Currently, our understanding of the disease mechanism is greatly enhanced, and over the years, several novel therapeutics has evolved to treat and manage the disease. However, the novel therapeutics developed are targeted towards certain forms of cancers, while there are yet many more which are in need of such strategies. In spite of these continually evolving therapeutics, cure for the disease has still remained elusive and it continues to pose a threat and challenge to mankind. One of the key reasons for this is the heterogeneity of the disease and diversity of the molecular mechanisms underlying the disease. It is clear the study of molecular biology that has given an insight into the disease mechanism, which has further aided in the development of novel drugs and therapies. Therefore, an understanding of the molecular mechanisms of the disease is of utmost importance to evolve newer strategies for efficient tackling of the disease and associated problems. Types of cancer. Cancers can be classified in various ways based on their clinical behavior, tissue of origin, or based on their mode of origin. Classification based on clinical behavior. Based on their clinical behavior, tumors can be classified into two types, viz. 1. Benign. 2. Malignant tumors. Clinically, the term cancer is specifically used to define tumors that have gained the ability to be invasive into adjacent normal tissue. Such tumors are called as malignant tumors. In addition, malignant tumor cells also acquire the capacity to separate from the parent tumor, enter the blood or lymphatic vessels, travel to distant places and form tumors in newer locations in the body. This property, or the ability to spread to distant sites away from the original site, is called metastasis. Malignancies that have attained the ability to metastasize are harder to treat and make disease management extremely difficult. Benign tumors. In contrast to malignant tumors, benign tumors remain localized at the primary site of their origin, i.e., remain clustered in a single mass. Tumors that remain benign can most often be completely cured, which is usually achieved by surgical removal. Hence, it can be said that the benign tumors constitute a less aggressive form, whereas malignant tumors are more aggressive forms while considering their clinical behavior. However, some benign growths can act as precursors for the formation of malignant cancers, e.g., colorectal and thyroid cancers. The characteristics of benign and malignant tumors are given below. Figure 4.66 Comparison between benign and malignant tumors. Benign tumors are well defined and composed of cells that closely resemble the tissue of origin, with a slow rate of division. Malignant tumors are irregular with abnormal cells, which have a high rate of proliferation and are invasive. Benign malignant. Encapsulated, i.e., defined by a clear boundary. Irregular, i.e., the distinction between the edge of the tumor and the surrounding tissue may be unclear. Closely resemble the normal cells, tissue from which they originated often do not resemble normal cell tissue from which they originated. Show a low rate of cell division, show a very high rate of cell division. Cell nuclei have a regular shape with a small nucleolus. Cell nuclei are irregular, large, with pronounced nuclei. Non-invasive, do not infiltrate into adjacent tissues. Invasive, infiltrate into adjacent tissues and may cause serious damage to the adjacent tissues. E.g., cancers of the epithelium destroy basement membranes and invade the stroma. Do not show metastasis, can show metastasis. Cells from the primary tumor can separate, migrate through blood or lymph to distant locations and establish secondary tumors. I.e., from their primary location, they can migrate to different organs and form secondary tumors. Usually can be resolved by surgery, however, sometimes can become complicated depending on the location, for e.g., brain. Outcomes are variable depending on the stage of malignancy. Table 4.8 Differences between benign and malignant tumors. Classification based on site of origin. Tumors can be classified into two types based on their site of origin, viz. 1. Primary tumors. 2. Secondary tumors. Secondary tumors. The tumor at its original site, e.g., breast, colon, etc., is called as the primary tumor. The tumors that are formed at other sites as a result of metastasis of the primary tumor are called secondary tumors. The secondary tumor can be found in any of the several other anatomical locations other than the original location in which the primary tumor was found. Classification based on cell tissue of origin. Based on the cell type or tissue of origin, cancers can be classified into the following major classes. They are: 1. Cancers of epithelial origin, e.g., carcinoma. 2. Non-epithelial cancers of sarcomas. B. Cancers of hematopoietic tissues, including cells of the immune system. C. Neuroectodermal cancers, cancers of the nervous system. Causes of cancers. The several steps in the development of cancer are each generally governed by multiple factors, such as genetic constitution, environment, lifestyle, etc. Although the exact causal factors for many of the cancers are unknown, certain factors have been identified for a few cancers. Studies involving large human populations are conducted to identify the incidence and prevalence of diseases such as cancer. Such studies are called as epidemiological studies. Epidemiological studies of cancers consider factors such as geographical distribution, lifestyle, diet, environment, etc. These studies help to gain insight into the etiological causal factors involved in cancers. Risk factors may be environmental, lifestyle-related or unavoidable, or genetic inherited, and are generally specific for particular cancers. Inherited factors are clearly of major importance. However, most cancers arise through a combination of genetic and environmental factors and less commonly by extreme exposure to radiation, viruses, cancer-causing chemicals, carcinogens, or combinations of genetic factors only. However, having a risk factor, or even several, does not guarantee that an individual will get the disease. Risk factors involved in cancer can be broadly classified into two categories. 1. Environmental factors, including lifestyle-related risk factors, such as smoking, unhealthy diet, or unprotected exposure to strong sunlight. B. Factors pertaining to the environment are not under the direct control of the individual, such as high levels of radiation, cancer-causing chemicals, and certain infections. C. Unavoidable natural processes, such as free radical generation, endogenous hormones, and cosmic rays. 2. Genetic factors, including inherited mutations, e.g., Li-Fraumeni syndrome due to a germline p53 mutation and genetic predisposition to various types of cancers. B. Acquired mutations. Sporadic cancers often occurs in combination with environmental factors. The genetic environment of certain individuals may make them relatively more vulnerable to develop cancer. Risk factors act in combination. It is often difficult to attribute tumorigenesis to any single causative agent, but exposure to specific environmental factors is strongly predictive of the future development of particular cancer. Tumorigenesis to any single causative agent, but exposure to specific environmental factors is strongly predictive of the future development of particular cancers. Particularly notable are the strong links between cigarette smoking and lung cancer and exposure to ultraviolet UV light and skin cancer. Genetic predisposition to cancer. Traditionally, the genetics of inherited cancer predisposition has been considered in terms of single, high-risk genes with high penetrances, such as in retinoblastoma RB, Li-Fraumeni p53, familial breast cancer BRCA1/2, familial colorectal cancer HNPCC, and Wilms tumor WT1. In fact, Knudson's hypothesis was first confirmed by the study of retinoblastoma. A list of the more common single-gene inherited cancer syndromes is given in Table 4.9. There are certain genetic conditions that make an individual more prone to developing cancer. Xeroderma pigmentosum, ataxia-telangiectasia, and Fanconi's anemia are examples of inherited gene mutations that attribute to cancer susceptibility. These inherited defects generally are characterized by deficiency in DNA repair pathways that are responsible for their cancer susceptibility. Other susceptibility genes include inherited tumor suppressor gene mutations, alterations in the metabolic enzymes involved in the metabolism of drugs and environmental toxins, and polymorphisms in genes regulating certain essential nutrient utilization, e.g., folic acid. Human Genome Project completion has allowed the discovery of genetic alterations that make individuals prone to developing various diseases via a systematic approach. A catalog of genetic variation involved in nutrient metabolism, catabolizing toxins, and DNA repair is being produced by the Environmental Genome Project. These data have been generated largely by analysis of single nucleotide polymorphisms (SNPs). This will enable the prediction of individual susceptibility to diseases promoted or triggered by diet, environmental pollutants, and other lifestyle factors. For example, SNP analysis has revealed that individuals with genes for altered folate metabolism show increased susceptibility to leukemia development after benzene exposure. Another example is the correlation between breast cancer susceptibility and ethnic variation in SNPs of BRCA1 gene. CLONED. GENE. Therefore, it can be deduced that the same selection process may also be functional in vivo during the progression of malignancy over time, from a lower grade to a higher grade. Decreased requirement for growth factors and decreased density-dependent inhibition of proliferation in culture or other properties that differentiate transformed cells from their non-transformed counterparts. Lower serum growth requirements are exhibited by cells transformed by oncogenic viruses when compared to normal cells. For example, 3T3 fibroblasts transformed by viruses such as SV40, murine sarcoma virus, polyoma, or Roux sarcoma virus are able to grow in a culture medium that is deficient have been observed, i.e., the appearance of new surface antigens, glycolipids, proteoglycans and mucins, as well as altered cell and cell ECM, extracellular matrix communication. Modification of extracellular matrix components. The ECM is involved significantly in regulation of cell proliferation and differentiation. It is now known that interaction with mesenchymal stroma on which tumors grow and development of blood supply plays an important role in growth, invasion and metastatic potential of tumors. Continuous remodeling of the supporting stromal structure takes place by interaction of the grown tumor with host mesenchymal cells and vasculature. Tumor cells can breach the basement membrane, a specialized type of ECM, barrier by release of a variety of proteases, collagenases and glycosidases that can degrade various matrix components. This allows invasion of tumor cells across tissue barriers, blood vessels, and lymph channel walls. Additionally, certain matrix components can be secreted by the malignant cells and possess receptors for certain components. This capacity is known to enable the binding of malignant cells to the vascular endothelium and is suggested to be involved in tumor metastasis.